Recording going. So welcome everyone to this month's seminar series from the Centers of Excellence in Iron and Heme Disorders that's sponsored by the University of Utah and NIDDK. Today we have the privilege of hearing from Tracey Rouault, who comes to us from the NIH, and she's going to speak to us on the role of iron sulfur proteins in mammalian cell heme synthesis and viral infections. I'm not going to spend a lot of time introducing Tracey because most everybody knows her, with the exception of saying Tracey is maybe one of the most prolific writers we have seen. We were talking about it in the group here that we all rely heavily on Tracey's reviews. So we want to just say thanks for that, Tracey, and bringing all the data together in a very coherent way. And we're really anxious to hear your presentation today. So thank you for your participation, and we'll turn the time over to you.
Okay, well, thank you, Diane, for that very nice introduction. I'm happy to have this opportunity to speak to this group, particularly because I have something to say about the heme biosynthetic pathway that we published in 2020, right at the beginning of the pandemic. And it hasn't been picked up on by most people, and I'm trying to get it a little more visibility because I think it matters. And I also think that Utah, with its special emphasis on the heme biosynthetic pathway, that maybe I can reach some people that we didn't reach at the beginning of the pandemic who perhaps didn't read our important contribution. So the title of my talk, I had picked it in September, saying mammalian cells and viral infections. And I realized that I'd left out heme, which was the reason that I wanted to speak in this series in the first place. I was trying to go very general because I didn't know in September what we'd be wanting to talk about in March. But here we are. And so let me see if I can. Okay, so just to give some background, there are two proteins in mammalian cells, iron regulatory proteins 1 and 2, that also are officially named ACO1 and IREB2. And they're homologous proteins that sense cytosolic iron levels and regulate expression of the target transcripts by binding to RNA stem loops that are known as iron responsive elements. And this has been work that has been contributed by a great many people. I put a review from 2005 here that I think captures some of the early first 15 years of work, but I couldn't really put all the references that would be relevant to this. But that point was of great interest because when we started working on it, we were working on trying to find the protein that was binding to RNA stem loops and regulating translation of ferritin. And ultimately, it turned out that what we were looking at was this protein IRP1 that has two different conformations, one of which binds a 4-iron 4-sulfur cluster. And when it does that, it acts as a cytosolic aconitase, interconverting citrate and isocitrate. The structure of that was solved in 2006, and it's shown here as a cartoon. And then the structure of it in its other conformation, in which it binds to IREs, is shown here, and that structure was also solved. And this is a cartoon of that. And what it showed was that upon losing this iron sulfur cluster, IRP1 would undergo a conformational change that then made it an ideal protein that would bind to the RNA stem loop. And I've shown the RNA stem loop here with the 5' end. It has a bulge C in the stem loop. And then 3 residues of the loop itself actually really reach in to aspects of the protein that aren't available in the fully folded protein to form very tight binding. And the result of that is, and the reason that I'm showing that is not that I don't think that a lot of people know this, it's that I've realized in reading reviews that a lot of people don't know it. And I've seen it, so many mistakes on this point that I want to just take this opportunity to restate the position, which is that ferritin mRNAs, both H and L, and 5 other transcripts that are known to us, like HIF2 alpha, erythroid ALAS, and a variety of others, have an IRE, the RNA stem loop, in the 5' end. And in cells that are iron depleted, IRPs, both of them, bind to the stem loop. And when they do that, they inhibit translation by preventing assembly of the ribosome. When they are not inhibited by binding of IRPs, what you get then is active translation of ferritin. This is translational regulation at its finest, and it's very important in ferritin, which is, as you look now through normalized tables of how many transcripts there are in various cells, ferritin is really very abundant compared to most other transcripts. And so if you're going to try to regulate its synthesis according to iron levels and try to prevent it from being made when there isn't iron that should be stored, this is a very good way to do it. Now, I mentioned that the iron sulfur was the key to this transformation in its activity between being an active aconitase and an IRE binder. And iron sulfur clusters come in a variety of shapes and stoichiometries. The most famous of them are the rhombic 2 iron 2 sulfur clusters, and then there are cubane 4 iron 4 sulfur clusters, much more complex clusters such as those that are found in nitrogenase. And these are one of the points that I want to make is that in mammalian cells, these have not been found anywhere near as often as they likely exist. And I think that the reason for that is that they have a tendency to be very oxygen sensitive. And when we do our usual benchtop purification, we destroy the cluster, and then with our buffers, we reconstitute it to a certain extent, but zinc occupies the site that was occupied by an iron sulfur protein and cluster. And therefore, I think a lot of proteins that may be classified as zinc proteins are in fact secretly iron sulfur proteins that we've missed because we haven't handled the protein right as we analyze it. So that would be one take-home point that I hope to succeed in making, is that don't be so sure about your zinc protein. And iron sulfur clusters also participate in enzymatic activity through Lewis acid base chemistry, and also they promote free radical formation. Many chemistry possibilities are made possible by the iron sulfur cofactor. Our question was, if the presence or absence of the iron sulfur cluster determines function, determining whether IRP1 binds IREs or functions as an aconitase, how are these clusters made? And we got a good lead from the work of Dennis Dean and his colleagues working on the E. coli operon, in which they identified proteins that are involved in obtaining sulfur for synthesis of the cluster, a scaffold protein known as ISCU, upon which the nascent cluster is synthesized. And also the operon handily contained also a chaperone and a co-chaperone. And a nice thing about bacteria is the way that they've organized things into operons so that everything that matters appears to be in one place. So you don't get disorganized about what the chaperones, for instance, do you need the chaperones? And I think the answer from bacteria and from many other studies is yes. And so the chaperones then participate in a step in which there is transfer of the nascent iron sulfur cluster to the substrate protein. And we've done a lot of work on this particular step, and I'm going to explain why. But the answer is that there is a great deal of specificity involved in the transfer, a choice of substrate, and this entire apparatus will not associate with a protein that doesn't really want an iron sulfur cofactor and hasn't shown by means of displaying a motif and some other features that it deserves to have one. So how are these clusters transferred specifically to recipient proteins? We found in SDHB, which has 3 iron sulfur clusters, we did a huge yeast-2 hybrid study and a great deal of work to try to find what the motifs or features were in SDHB that made it a recipient for receiving the iron sulfur cluster from the synthesis apparatus that I just showed to you. And after quite a bit of analysis of the data in different pieces, we finally found that there were motifs that we could identify, and LYR is one such, but others we've derived subsequently a consensus motif in which the first position would be one of these amino acids, the second would be a tyrosine or a phenylalanine, so a large hydrophobic, and the third would be an arginine or a lysine, so a positively charged residue. And there are 2 such motifs in SDHB, and then there's one in SDHAF1, which is the accessory factor that helps load the cluster. And so ultimately SDHB ends up with 3 iron sulfur clusters. We posited that these motifs can be used to search for unrecognized iron sulfur proteins since, as I said earlier, we think that so many have been missed, particularly in mammalian cells. And a virtue of the LYR interaction would be that it would allow for low affinity transient and modular interactions. And so a handoff rather than a permanent association of the chaperone-co-chaperone pair with the recipient. Another thing that we found is that in mammalian cells, that an iron sulfur assembly also takes place initially in the cytosol in parallel to mitochondrial assembly. This is different from the models that have been circulated, and that in the cytosol, these iron sulfur clusters, which are nascent, can be transferred directly to some proteins, but also transferred to the CIA complex, which is composed of CHOW1, MMS19, and FAM96B. And they seem to be particularly useful as a platform for providing iron sulfur cofactors to nucleic acid modifying proteins. And right about 4 or 5 years ago, we had gotten to the point where we were interested in saying, if all work is going to be done on a bench with 21% oxygen, and then evaluate cells are going to be grown in incubators at 21% oxygen, how will we ever figure out what is really an iron sulfur protein in the cells? And we got interested in the idea that we could predict based on looking at motifs and whether something would be an iron sulfur protein. And we were also interested in the fact that we thought that there was a missing iron sulfur cofactor dependent protein in the heme biosynthetic pathway. And we had a number of reasons for thinking that. So we looked through the many heme biosynthetic enzymes, and we found a very good candidate, which is ALAD, also known as porphobilinogen synthase. And we, in fact, did find an iron sulfur cluster there, and I'm going to tell you that story, which is published, but I really don't think that hardly anybody has read the paper. So it'll be new to most people. Here's a schematic of the heme biosynthetic pathway. And as most people know, the first step takes place in the mitochondrial matrix. And then there are a number of steps that take place in the cytosol, shown here. The first being aminolevulinic acid dehydratase, the one that I'm going to talk about most here. And then the final steps are proposed to take place back in the matrix with ferrochelatase supplying the iron to the protoporphyrin ring. But in fact, there are actually 3 places at which heme synthesis is regulated by iron. And one is in erythroid cells, the ALAS that makes this compound is regulated by the same way that ferritin is regulated by IRP binding to an IRE and preventing its translation. That's a good check on heme biosynthesis because you don't want to make heme if you don't have enough iron, and not having enough iron is reflected in IRP's binding to the IRE. Then the second step, which I'm about to tell you about, is that the ALAD is an octamer in which each of the monomeric subunits binds a 4-iron 4-sulfur cluster. And then finally, most people are aware that ferrochelatase is a dimer that contains 2 iron sulfur clusters in mammalian cells. So that would amount to actually 3 checkpoints for manufacture of heme according to iron levels, which seems appropriate because one of the very bad things that can occur in this pathway is that these porphyrins can be quite toxic, and you don't want them to build up. But they would build up if you let these early steps go, or if you let the final step not supply the iron that turns the protoporphyrin IX into heme. And so I think 3 checkpoints in this pathway. We've added one more with the ALAD, and I think it's a good one because it prevents all these intermediates that are toxic from being formed. ALAD catalyzes the condensation of 2 ALA molecules into porphobilinogen. And here is the reaction. Here's ALA and ALA, and they condense to form porphobilinogen. And then that goes on to participate in many other larger, more complex molecules. And we were looking at ALAD when we were thinking about whether it would be an iron sulfur protein. And we used homology to show that the cysteines, some of which have been implicated in binding zinc in the form of ALAD that people have been working on for 30 or 40 years, and also a motif AFR shown here, which has the features that we look for, which is the F, which is the large hydrophobic in the center, and the R, which is the positively charged amino acid that fits into a pocket in the recipient proteins that are that want an iron sulfur cluster. Particularly, we found that the presence of an LYR type motif in conjunction with a metal binding site has predictive value. And in particular, in this instance, looking at ALAD, ALAD had been treated and viewed as a zinc binding protein for its entire existence as a studied protein. We looked at ALAD to find out whether it interacted with HSC20, which is the co-chaperone that we think confers specificity towards the recipient proteins. And we saw that ALAD, in fact, did co-precipitate with HSC20. And that to us is a very good screening maneuver to see if there's an endogenous interaction. We then went on from there and did a whole variety of things that are summarized in this slide, but also in the paper that we wrote in 2020. And basically, we showed that if we mutagenized the AFR motif, we would get no co-precipitation of the cytosolic HSC20, which you can see here migrates differently from the mitochondrial HSC20 because it's larger. And then we found that if we mutagenized the AFR, we inhibited incorporation of iron. We could see a UV absorbance in the wild type at around 420, which is characteristic of iron sulfur clusters, and we could eliminate it if we mutagenized the AFR motif. And then I'm going to come back to the Mossbauer. We overexpressed this protein in E. coli along with the operon for the bacterial insertion of iron sulfur clusters. And we got brown protein, which is shown here. And this color of sort of tan brown with a slight almost red hue is very characteristic of what these clusters look like in solution. And then in collaboration with Carson Krebs and Marty Bollinger and those at Penn State, we did Mossbauer spec on these. And Mossbauer really is a great equalizer in a way. There isn't much, there are very few ways to fake it. And it tells you a whole lot about the iron sulfur cluster. And in this case, we learned that we had a cubane 4 iron 4 sulfur cluster. And you don't get anything whatsoever because this depends on magnetism and so on and so forth, and a special machine and a lot of expertise. We wouldn't have this if there weren't an iron sulfur cluster there. So it false positives in this type of experiment are really rare, particularly if you're working with people who are honestly fitting their curves. And that led then to the conclusion that here we have ALAD is known to form an octamer, and we suggest that each of the monomers contains a 4 iron 4 sulfur cluster, and that is a checkpoint early in the heme biosynthetic pathway. We think that this offers new potential for insights into ALAD enzymatic activity. The identification of an iron sulfur cluster that assigns a position that was formerly assigned to zinc could facilitate the incomplete understanding of the enzymology, which is where things do stand as far as I know. I have not seen any publications for a while. And as I mentioned before, dependence on iron at the second step of heme biosynthesis could prevent synthesis of toxic porphyrin intermediates. If you look at ALAD, one monomer has a hydrophobic pocket into which several lysines stick, and they form a Schiff base with one of the ALA molecules. Here is ALA side by side with the lysines binding it. And here is how it's been modeled with zinc, which is bound to 3 cysteines. And this is the crystal structure of that, with the zinc shown here in green. And the point that I want to make here is that an iron sulfur cluster could easily occupy that same cysteine binding site. And in so doing, it could be a better platform for the condensation of these 2 ALAs, because it could easily bind the oxygens and the nitrogen from the ALA. And here again, here's an illustration from a conatase, which might function in a similar manner, in which the iron sulfur cluster is bound by 3 cysteines, and it has a 4th iron that's free to bind both oxygen of water and also the oxygens of the substrate, citrate or isocitrate. As I mentioned, no recent work has been published on the enzymatic mechanism. And we raised the question, was it not completed because the physiologically correct metal ligand was not being used? And there is another point, which is that an iron sulfur cluster would lead to a slightly different conformation than a zinc because it's larger. And so the shape and distances in the active site might be changed if you solve the structure with the cluster in place. And so iron sulfur proteins, they likely enable those proteins to tap into cellular reducing power to access energy and sense metabolic status. And after feeling encouraged by the work we did with ALAD, we were ready to try again for another target. And at that point, we had the onset of the SARS-CoV-2 pandemic. And that naturally led to us searching through the open reading frames of all the proteins that encode the various components of the SARS-CoV-2. And ultimately, we found the kinds of signs we were looking for, which is LYR motifs and zinc binding sites, because so much work had been done on the structure. And we found 2 cubane iron sulfur clusters in the replicase NSP12 in the position we've shown by mutagenesis that zinc was thought to occupy. And we published that, and it was really rather unexpected that a virus would incorporate the iron sulfur cofactors. Then in the helicase, NSP13, which is involved in template unwinding, we found a single 4 iron 4 sulfur cluster. Interestingly, about all of these iron sulfur clusters, they're ligated by 3 cysteines and 1 histidine, which is noteworthy. And we found that the stable nitroxide temple, which is a stable free radical that can attack an iron sulfur cluster, inhibited the replication of SARS-CoV-2 in culture by degrading the iron sulfur cluster. And it also inhibited the growth of the related coronaviruses, SARS-1 and MERS-1. And it also mitigated disease in COVID-19 infected hamsters. So we could translate this to an animal model. At the time that we got this out, Paxlovid had already arrived in the market, and we were not really able to pursue this as a potential treatment, although I think the day will come when we'll be able to make more of this. The structure of the complex of NSP12 replicases with the NSP13 is shown here, and it's based on a PDB that was deposited. And you can see relative to the RNA here that we have an iron sulfur cluster here, and we have two here. And when we look at the alignments of the NSP12, all of the residues that are important for ligating the cluster are conserved even in the coronaviruses that cause merely a common cold. And here is another picture of their ligation, again, drawing your attention to the fact that in each case, one histidine is involved in addition to the 3 cysteines. And then in NSP13, again, we saw high conservation of the residues that are important, and we found an LYK motif, which would be a motif that would interact with HSE20. And interestingly, it was in a region known as the zinc binding domain, and we've concluded that it binds 2 zincs and 1 4-iron 4-sulfur cluster. And we concluded that through mutagenesis and through ICP-MS, in which the stoichiometry was really, extremely tight. And here's the LYK motif that permits the iron sulfur cluster to be inserted. If we take MERS-CoV, MERS is the Middle Eastern respiratory virus that is almost as bad as SARS-CoV-2, but the intermediary is camels, and so we don't get it here in North America. I think it's a good thing because it has a really bad reputation in places where it's you can get it. HCoV OC43 causes a common cold, and I'm going to tell you about this, but we're working with this in the lab because we wanted a model that would have all the retained features of SARS-CoV-2, except that it wouldn't cause a deadly respiratory illness in people who are working with it. I think that's a real selling point. And so here we're looking at the ability to do a primer extension of MERS-CoV-2, OC43, and SARS-CoV, and each of them has the ability to elongate an RNA primer, and each of them is dependent on an iron sulfur cofactor for its ability to do that procedure. I'm back to the structure of the whole polymerase complex, and we're questioning what roles these iron sulfur cofactors play. And for that, we really need models of the replication motion, structural studies that using components of OC43 would be feasible for our needs. And a question that we have that we're trying to address now, but it's not entirely straightforward, is do these iron sulfur cofactors enable viruses in infected cells to divert reducing equivalents that normally maintain homeostasis in host cells into the work of replication? And here is a hypothetical structure. I'm not going to talk about this at all. It's unpublished, but we found iron sulfur clusters in several more of the cofactors of the multimeric replicase complex. They're shown here just fancifully placed. And I'm suggesting that perhaps they play a role in electron tunneling, slightly analogous to that which we see in complex 1, where movement of the electrons is associated with energy capture. Pathways in which iron sulfur proteins may be critical. We know about the citric acid cycle in the respiratory chain already. A lot of tRNA synthetases many DNA metabolism proteins, translational initiation and elongation, ribosomal biogenesis and function, and untold other pathways that really haven't been examined with an eye towards finding a point at which an iron sulfur cluster would make the difference. And then I add to this viral infections from 2021 on. Prior to this, and our work on SARS-CoV-2, it was not something that was really considered very deeply. But replication really is the central Achilles heel in viral infections. If you look at what it takes all of them to get their job done. And we found in searching the literature that multiple other viral families depend on iron sulfur cofactors. There were miscellaneous reports about the rotavirus NSP5, Merkel cell polyomavirus, HBx protein from hepatitis B, our work, and some more recent work on a group of viruses called megavirinae giant viruses. We then predict that this is going to be a common theme in viral infections in general, because with the smallest genome possible, they take advantage of anything that the mammalian cell has to offer it. And the mammalian cell has a very functional iron sulfur cofactor synthetic pathway. And so it does make sense, since these are so useful in all the chemical interactions that a virus needs to produce, that it would take advantage of what the eukaryotic cell is up to and use the iron sulfur clusters that are being made for its own purposes. So I'd like to stop there and thank my colleagues. Some of this work, Gang Liu, who did the ALAD work, is now in China, and I don't have a picture of him. Nunziato Mayo did the work that I talked about on the SARS-CoV-2 and helped a lot with a lot of the other things that I said. Bernard Lafont and people in NIAID who had a SARS-CoV-2 special lab. Our colleagues at Penn State University, Debong Cecile, Marty Bollinger, Karsten Krebs, who did the Mossbauer, gave us the confidence about the iron sulfur cofactor. In the urologic oncology branch, they gave us access to Temple. And so I think in summary, my message would be an iron sulfur cofactor is really key in heme biosynthesis in a place where it was not expected. It may lead to other insights, and that iron sulfur cofactors are probably a great deal more common in many mammalian proteins, and we need to look for them both from an informatic standpoint and to improve our techniques such that we don't destroy these very sensitive cofactors in the process of trying to characterize them. So I'd like to stop there and take any questions.
Great talk, Tracey. Thank you so much. I've written in the chat, if you want to ask questions, feel free to unmute yourself and join in the discussion. Yeah, speak. So we have a postdoc from Montana State University from Jennifer Dubois' lab who has a question for you.
Okay.
The first question is that in the first step of the heme biosynthesis, is it disconnected?
No, I don't know what it's doing. Jumping around. Keep going.
Okay. So there is in the respective protein, there is a replacement of the iron 4 sulfur 4 cuban that replaces the zinc. So the first question is that how does the replacement happen? And second question is that when the zinc interacts with ALA, it may have acts as a Lewis acid. So in place of when it is replaced by the iron sulfur cluster, does it act in the similar way? Because iron sulfur cluster, as we know, usually acts as an electron transfer. So in this case, is it acting as a Lewis acid? So these two
Well,
questions.
that's what I would like one of you or your colleagues to figure out. I think it could easily be a Lewis acid because that's how it works in a conatase where there's a 4 iron 4 sulfur cluster. So it's far from unprecedented. You asked me, how does the iron sulfur cluster replace zinc?
Yeah.
My point is that, and you seem to frame it as though the iron sulfur cluster was pushing out zinc. But according to what I'm saying, as the protein is folding and undergoing its first assembly, it interacts with the iron sulfur cluster assembly machinery. And the first thing that happens is that an iron sulfur cluster is inserted into that site. And so there's no displacement. What happens when we ultimately work on some of these proteins is that through oxygen exposure and rough handling, we've degraded that iron sulfur cluster. And then from the buffer, zinc can come in and occupy the cysteonyl ligandiene space. So in fact, we have an experiment in our paper that I wish you'd take a look at, because we tried, we can overexpress these proteins in eukaryotic cells, HEC293 or XP293. And so in that setting, we're asking the protein to just rely on the mammalian ability to know what's right. And we tried overloading those cells with zinc to try to force zinc into that site in an intact cell that was functioning with all of its pathways doing the right thing. And we couldn't get it to put zinc into that site in the cell. What it ended up was making more proteins that are zinc metallochaperones. So we got a huge increase in those, but no zinc in the active site. So the point then is that the delivery, because of the LYR motif and the specificity, there's no confusion when you're in the cell about whether zinc belongs there or protein. It's not an accident. It's a specific delivery by the apparatus of the cofactor that protein needs. Does that help?
Yeah.
Thank you. Ova, do you want to ask your question or follow up?
Yeah, I wanted to follow it up, Tracy. Is there any difference in this ability to replace or displace the iron sulfur cluster with zinc, and vice versa, when you have a cluster made of 4 cysteines rather than 4 cysteines and 1 histidine, due to the fact that the zinc has, after all, some affinity for histidine as well?
Well, let's see. So one thing that I'm saying is that it's a one-way street. We don't, unless you're working on the bench top, which would be different, that the iron sulfur goes in first and only. So you can't displace it with zinc until you've gotten it out of the cell and onto the bench top and harmed it, attacked it with an oxygen radical or something of that nature. As to whether the cluster ligated by 4 cysteines versus 3 cysteines and a histidine is more readily decomposed by oxygen or not. I don't know that we're in a position to make a generalization like that at this point. I guess my position is no comment. I think all the iron sulfur clusters are to varying degrees able to be destroyed a lot more easily than anybody supposes.
Thank you.
Okay, we have another question from Namita Kumari.
Yeah, so this heme has been known to inhibit a couple of viruses like HIV, Zika, Ebola, and all that. So how do you place this iron sulfur protein in relation to heme or anything like, do you feel that you are suggesting that they might, the viruses might use them as a target for, for their benefit, whereas heme kind of inhibits it. So, can you put it in context? Like, I did not get, you have in your title that, for heme synthesis also, right? So I don't know if I'm making it.
Well, I'm not sure if I actually caught your exact point. In fact, I'm pretty sure I didn't. So
heme, can I put it bluntly? So heme on one hand kind of inhibits the viral replications, like couple of viruses, like
Mhm.
including SARS also, I guess,
mhm.
right? And this iron sulfur cluster will be kind of used by the viruses for their benefit.
Yes.
So
Well,
in that way, they are opposing. So like will inhibiting the this is kind of this FES will also if it is iron is available in proper quantity, heme synthesis would go on, right? In that way, the viruses would be inhibited. I'm thinking on that lines.
well, I can only really speak to a couple of viral infections, and our impression is that when the virus goes into its high growth phase, that it really takes over the protein synthesis machinery and a whole lot of other machineries, and that most of the new proteins that are made, or a really large percentage, are viral proteins, and most of the iron sulfur clusters that are made by the eukaryotic cell are immediately taken by the virus. So there isn't much left over. So I don't think in an acute viral infection that much heme is likely being made, but I'm saying that on the basis of SARS-CoV-2 and OC43. So something more chronic, I don't have information that would really speak to that.
Okay, thank you.
Any other questions? Yeah, Ella, go ahead.
Hey, it's sort of a follow-up on this thing that you were just discussing. So if you want to treat viral infection with something like Tempol, you may disrupt also host iron sulfur cluster assembly and function. But if I'm interpreting right, what you're just saying is that at the time of viral infection, the virus is hijacking most of host machinery anyway. So host machinery is disrupted anyway by the viral infection. Therefore, you would likely mostly target just the viral infection when you interfere with iron sulfur clusters. But how about the cells that are not infected yet?
Well, what you said is exactly what we think. Another thing that we think is that a lot of these protein complexes that depend on iron sulfur clusters are, if they're already made before the virus strikes, that they're actually fairly stable. And so you can get through the 5 days and still come out if assuming that the patient or animal survives with function. So it's this massive hijacking that is impressive, and I think roots the temporal effect towards the virus.
Thank you.
Other questions? Yeah, we have one here in the room. So this is from Kay Loveridge, who's in Paul Segala's lab.
Yeah, thanks for that super interesting talk. I had a quick question about the LYR motif, and I apologize if I missed this, but right, I guess my understanding of LYR proteins is there's both that LYR motif and also like an important 3 helical bundle structure that goes to their function with iron sulfur clusters. So I guess like specifically when I saw the NSP13 protein, like the cartoon you showed, I didn't necessarily see any evidence of that 3 helical bundle. Is that also important when looking for these LYR proteins, or do you think it is just the motif? Yeah, I'd just love to get some of your thoughts on that.
Okay, I think you're getting into something in which the informaticians had annotated an LYR motif family, and the definition of them was that they would contain an LYR motif within the first 20 amino acids, and they said a few more things. We don't limit ourselves to the first 20 amino acids by any means, and also no structural limitations. And part of that is that most likely it is that as a protein is being translated, that the LYR motif can interact with the machinery that transfers the cluster, and before it's even completely translated, and that would be a reason that the cluster can drive conformation. And so, for instance, in SDHB, if you look at the fully folded protein, most of the positions of those clusters, once it's fully folded, you can't come from the outside and insert an iron sulfur cluster. It has to be there and have the protein fold around it. And there were studies done decades ago in which people took a protein that is an iron sulfur protein like ferredoxin and moved the cysteine ligands around. And what they found when they solved the crystal structure was that they had a different structure. So ligating an iron sulfur cluster drives conformation, and we don't really believe that a preset conformation has a limiting role in what can be inserted. Does that help?
Yeah, that does. Thank you so much.
A couple more comments in the chat. Carolyn Philpott, do you want to go ahead and unmute and ask your question?
I would just like to hear what Tracey has to think about the significance of this 3-cysteine, 1-histidine coordination environment. And if anybody else in the audience has comments along those lines.
Well, here's what I think based on talking to people who have thought about this quite a bit more, which is that a 3-cis 1-his coordination is more likely to support electron transfer because the His almost opens up a spigot on one of the vertices of the cluster. And it can also change its reduction potential a bit. So I went to the Metals in Biology Gordon Conference and showed this to quite a few people who had opinions. And it tended to be that it really looked as though these clusters were all more likely than not participating in some type of electron transfer rather than some of the other options, like the 3-cysteine ligation of aconitase, in which it becomes a Lewis acid.
Okay, thank you. We have another question in the chat from Lorena. You want to unmute yourself?
Yes, thank you. So it's kind of a follow-up. Do you think that 3-cysteine 1-histidine structure will make those proteins to be more susceptible to tempo, for example?
I'm not sure. The other example that I know really quite well is aconitase, which is 3 cysteines and no 4th ligand. And that's extremely sensitive to Tempol. We can give Tempol an animal feed and feed animals for 18 months. And the major change that we see is the change in cytosolic aconitase. I think that a 4-cysteine ligand situation would also be susceptible to Tempol, but I can't offer an exact example right now.
If I can follow up on what Tracey said, Tina. So I think we have been discussing the significance of the 3-cysteine 1-histidine coordination, and something that we found in the literature that's been experimentally validated is the case of nitrogenase. And people have done a mutagenesis of the 4th ligand, the histidine into cysteine. So the 3-cysteine 1-histidine coordination favors 1,000-fold faster electron transfer than coordination by 4 cysteines. So the thiolate group on the cysteine seems to essentially slow down electron transfer. So it's been proposed that a 3-cysteine 1-histidine is very common in large multiprotein complexes which have more than one center that can relay electrons. So that's, like I said, the only evidence we have is the example of nitrogenase, where replacing the histidine with the cysteine blocks electron transfer by orders of magnitude. And we also think that, I mean, as well known, the environment of the cluster essentially affects the reduction potential of the cofactors. And so it can make them more or less susceptible to Tempol, which is a mild oxidant. So something to keep in mind is that, like Tracey said, most clusters are buried, but also the reduction potentials is going to essentially dictate whether they can be oxidized by a molecule like Tempol, which is not a very strong oxidant.
Thanks. Good intel.
There's a comment and question, I think, about mitoneate and redox regulation about the 3-cysteine 1-histidine like mitoneate. I don't know if anybody who's populated those questions wants to clarify what they're asking.
There is a paper on that, right? So it was found that the mitoneate with the 3-cysteine 1-histidine had a given reduction potential, and replacing the histidine with the cysteine was unable to essentially modulate the potential of the protein. So there is a paper, I think, from Sean Elliott that shows a reduction in the reduction potential by 100 millivolts upon binding of pioglitazone, one of the molecules that are known to be bound by mitoneate. Replacing the histidine with a cysteine block that transition in the reduction potential. So I think, like I said, it might be just the environment and the ability of histidine to be protonated, which is something the thiolate group is unable to do.
Thank you.
Yeah,
Thank
there
you.
is another issue there, which is the stability also of the cluster. I think it's been shown by the same group, I mean, of Rahel Nehustai. I was part of that, so I know this very well. So that, the question of the stability also has to do with that. And I was referring to that when I asked also Tracey, to what extent also changing slightly the acid environment there may affect also the stability of this cluster. And the histidine seems to confer some lability to the cluster. It could be, just an experimental artifact, but the question is whether something like that exists also in vivo.
Tracey,
Well-
do you want to take that?
Well, in a sense, Yoav, we're working on those questions, and we're working with Sean Elliott. We've gone straight to the source. So we hope to have some answers of our own.
Great.
I'm gonna stop the questions there. I hope you guys will reach out to each other some more. Tracey, what a great talk. Lots of thought-provoking ideas, and the idea of more iron sulfur clusters as a normal population in proteins is really great. It's fun to think about. So
Well,
thank you. And thanks for everybody's participation. For those of you who want to hear this again, the recording will be posted on our YouTube channel, which you can find on the CIHD website. You like that? We have a YouTube channel. But it gives you a chance to go watch some of these previous seminars if you've missed them and learn a lot more about what's going on around the country and the world in iron homeostasis, heme homeostasis. So take care, everybody. Thanks for dropping in, and thanks for the wonderful presentation.
Oh, thank you very much.
Thank you.
Everybody, remember, I'm recruiting.
Hey, Tracey, if you want to send me a recruiting announcement, I can post it on that website as well.
Okay, thank you.
This is Joe Prikal. Can I ask you something unrelated?
Do it.
Yeah. I'm commenting on your blood paper 2 years ago when you talked about deletion of iron responsive protein 1, and you had an animal model and studied erythrocytosis. And I heard that you also studied some human patients, but I have not seen it published. Are there any patients with IRP1 mutations who have erythrocytosis?
Yes, they were published by the Icelandic group.
No, I saw there was a whole genome and looking at the, but clear-cut phenotype of autosomal dominant inheritance in multiple generations. Are there papers?
Yes, it has been published. It was by the senior author was Carey, the Icelandic genome guy. And he had pedigrees that went back 700 years through the Icelandic churches. He had erythrocytosis, and he had 2 types of IRP1 mutations. He had those that caused anemia and those that caused erythrocytosis. And I have it quoted in a paper. We have a paper in Seminars in Hematology in 2021. Carrie Stephenson. That's
Yeah,
it.
okay, right. Thank you so much. Somehow I missed it. I appreciate that.
Well, I mean, people have missed a lot. We're trying to regain our footing after the pandemic.
Oh,
that's got it.
Okay, thank you all for
Great
your
job.
participation, and wish you all the best.
Yeah.
Thanks,
Thank
Tracey.
you.
Oh, bye.
Bye.
Bye-bye.